hear me? Okay, first I want to thank the students for the opportunity to come here today, and also thank Talbray and Pioneer for sponsoring this event and all these series. It's a great activity. So today I'm going to talk about basically how I'm laying out my breeding program. I thought this might be relevant. I've only been here for about a year or less than a year, so I don't have too much data to share from my time at LSU, but I thought maybe some of the students in the audience would be in a similar situation in the coming years of starting a new breeding program and kind of thinking about how they want to structure that, where to begin, what kind of questions to ask. So I'm going to talk a bit about that, at least the way I've gone. So to outline my talk, give a little overview of rice in the United States, Louisiana, kind of the structure of the established breeding program there at LSU, and some of the opportunities we think that markers could play a beneficial impact in the program. And then lastly, I'll talk about some of the logistical considerations of how to integrate these new technologies into the established pipeline. First, I'll talk about the research station. I work at the H. Rouse Caffey Rice Research Station. This is part of the LSU Ag Center. This is be here, like the equivalent of Geneva. It's off-site, it's off the main campus. It's about an hour and a half west of the main campus in Baton Rouge. This station, I believe, is the oldest rice station in the United States. Founded over 100 years ago. From the beginning, it's been involved in variety development, evaluation. So there's been over 50 varieties released since the opening there. One notable achievement was the development of the Clearfield technology, herbicide rice resistance. This was later licensed to BASF, but this was discovered at the rice station. Currently, we have 50 full-time staff, including faculty and all the research associates. And the education, or the research and extension is the main focus. We do some education, have graduate students, but the main focus is extension and applied research. So you can see the general areas that we have research activities in. So I want to just put this up here. This is historical rice yields in Louisiana, and I want to show this for Louisiana is one. A lot of times I hear these talks, and just last week at a conference in Texas, people talk about the yield plateau and all the things we need to do. Well, we haven't experienced that yet in Louisiana. Yields have been constantly on the rise, and then this is a graph I took from a publication for all the other states as well. So you see Louisiana is this yellow line. The rate of increase has been higher here. A lot of that is attributed to the Clearfield technology. It changed how we can do rice farming because it gave us a herbicide to control one of our biggest weeds. So all the states have good increases in yield. So at least rice in the United States, we certainly are experiencing a yield plateau. This is a pedigree kind of view of all the varieties that have come out of the LSU program. The ones in, don't try to read them obviously, but the ones in green are varieties that were released, and the ones in orange are kind of founders, so new germplasm that came in. And the thing I want to point out with this is, you see, it's pretty closed. The varieties that end up making it to the market typically come from crosses that were made from other varieties that were on the market. So that's not to say we don't make a lot of crosses with other things, or in the past haven't tried to make crosses with other things. It's after you go through the years and years of testing, what ends up coming out are the crosses that were made by the elite things. So again, I just want to make that comment here is that the program is highly focused on elite by elite crosses. And based on those two previous slides, that has been successful, and we continue to get very good gains. So there's three main classes that we breed for. The first is long grain, and that's here. That's probably 75% of our activity is in long grain rice. And then we have medium grain, which falls in, here that kind of looks like a profile face, I didn't realize that, but that's coincidence. These are the medium grains. So again, you see some crossing between, but for the most part, the medium grains are crossed among the medium grains. We typically will cross across the long grains and the medium grains if there's a specific trait. So for example, Clearfield originated in the long grains. When we introduce that into the medium grains, that we have to do a cross between those groups. And then the last is the aromatic. This is a specialty rice market in Louisiana and the U.S. Very small acreage. So these would be, jasmine type aromatic rices. All right, so a little bit on just rice production in the United States and the market. Rice is produced in the South in 5 states. Up here along the Mississippi Delta, a little bit in southeast Missouri. Arkansas is the largest producer in the Delta here in Louisiana and Mississippi. And then our main region in Louisiana is where our station is southwest Louisiana. This is pretty much all rice farming there. And then along the Gulf Coast in Texas. So this is pretty much one production area, and then California has its own production area. It's kind of separate. It's hard to move germplasm for phyto reasons between the regions. So last year acres was down a little. It was around 2.7 million acres, I believe. We produced 9 million tons of rice in the United States. 85% of the rice consumed in the U.S. was produced in the U.S. But the main market for a lot of our rice production in the United States is export. So over 50% of the rice in the U.S. is exported to over 100 countries. So it actually makes the U.S. one of the largest exporters of rice globally. Certainly not one of the largest producers, but we export a lot. Most countries don't export anything. So the export market is a very important piece for us, and something that we pay very close attention to in our breeding efforts. It's the theme of this talk, as the export markets are demanding higher quality rice as people in many countries move to cities. It's harder and harder to get them to produce, to eat rice. It's harder to prepare than a lot of other foods that you can buy packaged. So Japan and Korea, this is an issue. And as people in other countries are asking for higher quality rice, and it's a bigger and bigger demand, so more and more effort is being put on it. So now I'll just focus a little bit on Louisiana. This year we had just over 400,000 acres. That's down. Typically we're around 450 or 500,000 acres. Last year we had 83% long grain, 15% of the medium grain, and 2% of the specialty kind of aromatic rices. What I'll point out here is different than some crops is the large majority of the varieties planted in Louisiana by Louisiana producers, and actually across the South in general, come from our program or from the public program. So there's not a large industry presence in rice. There is Rice Tech, who produces hybrid seed, which is very high yielding. So they do have some of the market, but there's 2 or 3. I listed all the varieties that were over 5% of the acreage. So there's 2 or 3 Rice Tech hybrids on here, but the rest are varieties out of our research station. So what we're producing is being grown by farmers. So our focus is very much on what is going to benefit the farmers and the end users. 60% of the acreage in Louisiana is clear field, so that's that herbicide-resistant rice. It's now owned by BASF. And 25% of our acreage is hybrid. So that 25% at this time would be Rice Tech hybrids, and then 75% would be LSU varieties is what's being grown. This past year, our averages were down quite a bit, but we had an excellent ratoon crop, and I'll talk about the ratoon crop in a little bit. But basically, along the Gulf Coast, we have a long enough growing season where we can get 2 cuts. So most of the numbers reported here are just always from the first cut. I don't know why they don't add them together, but you cut it once in the end of July, and it'll grow back, and you cut it again in middle or early November before we get a frost, and you get a second crop. So this past year, our first crop was down, but often the second crop will make up for it. So when you add them together, it basically came out to be a pretty average year. So there's a little bit of flexibility in there. The yields were down because we had a really wet spring. It's hard to get into the field and fertilize and things like that. So the ratoon crop this year was 50% of what the first cut was. So it's a very nice advantage of being down there. Texas and southwest Louisiana are the only environments that we can do that in. The other thing, I just put this in here because I found it interesting, is crawfish. So the rotation crop in southwest Louisiana is crawfish. This is a big crop down here. There's actually a lot of farmers now, rice prices are not favorable, are making more of their income off of the crawfish. So it's a complete aquaculture system. So last year we had over 200,000 acres in crawfish production. The way this works is, let's say last year or this year you're growing rice. Once you put that permanent flood on in July or June, you'll seed it. So you'll dump a sack of crawfish in there. Those crawfish, while the rice is still growing, will grow. They'll start to reproduce. You'll cut your rice, and those crawfish will reproduce. You'll let them grow for a few months, and then this is now crawfish season. So January to July, they'll go in here and harvest these. So the fuel for this is the rice stubble. So when we cut the rice with a combine, we'll leave that stubble there, and that is what is the main forage to sustain the crawfish. So these are just some pictures. This would be in the middle of your rice season. You might take some crawfish from your neighboring pond. Like again, this is a rotation, so some of your fields would be in rice and some would be in crawfish, and you would switch. So this gentleman is seeding this rice field with some crawfish, which will be harvested the subsequent year. These crawfish will grow and reproduce. They'll burrow into the ground, and then that's, I think, where a lot of the incubation or the hatching happens. And they'll come back in, and this is a field. So this would be a field that's in current production. You can see some of the rice still sticking up, but those little dots you see on there are the traps. So we'll go in. It's very manual. There's not that much mechanization in this because it's concentrated Southwest Louisiana. So they'll go through in this little crawfish boat and pull the traps and bring them. And it's primarily in the South. Louisiana, Texas is a big market for it. And I guess as you radiate out from Louisiana, it gets less and less. But you can go online and buy them and have them shipped up here overnight. And then you boil them and you eat the tails, and it's usually in a net. So anyway, I just thought it was interesting. So I'm going to shift gears a little bit here and talk about our breeding program and our targets. So I mentioned, we're really focused on supporting the growers of the state of Louisiana. And there's two main targets that people care about. It's yield and it's quality. So a lot of crops, maybe corn, primarily it's yield, and all the other things are secondary. For us, quality is just as important, and it's becoming more and more important. So these are two difficult traits to breed for. So we got to find a balance to how do we make progress in one and not shift too far. So quality is this is an example of chalk. This would be an appearance trait. This would be bad, this would be good. So grain quality is a complicated topic because it really depends on who you are speaking with. So grain quality, all these users will use that term grain quality, but depending on what circle you're in, it's a different trait that we're actually interested in. So we might say, great, we're only interested in two traits, yield and grain quality. It's not really two traits. Grain quality is many traits underneath that umbrella. So you have the farmers, millers, processors, that might be producers, Kellogg's or something like that, making Rice Krispies. So Kellogg's takes the medium grain rice for those Rice Krispies. If Kellogg's doesn't improve a medium grain variety, it pretty much kills that variety because there's not a market for it. So we need to try to consider all these variables of what is quality to people. So the traits in general, uniformity is a big one. So in a harvest, are all your seeds the same size? Are they maturing at the same time? Moisture, that's very important for a lot of these downstream things. We focus a lot on milling, which would be your total head rice. So if you have 100 grams of seed, how many are left after you mill it? And how many are broken? That's a very big one. This is one of the biggest bottlenecks in our breeding program is collecting these samples. You have to do it by hand, you have to process them, you have to weigh them. Others are grain dimensions, size and shape, appearance, so that chalk I showed on the previous slide, the color of the grain, aroma, how it cooks, how it smells, and then some of these chemistry traits, such as the amylose content, which is the texture. So if you have like a Japanese or sushi rice, it's a very low amylose, it's very sticky, and then some of the other rices were more intermediate amylose, it's less sticky. And then some of these other gel consistency, how it settles after it cooks, gel temp, how long it takes to cook. So these are all things that we need to consider. I will point out, luckily, the breeders over the last 100 years have done a very good job in basically fixing some of these traits within the elite germplasm. And I say this again to go back to why we end up always back with the same crosses is because if we crossed outside of those two crosses, all the progress that was made in these areas is now you have to select on it again. So it's very, very difficult to go outside and recover something that's going to meet all these criteria. So for example, in a long grain by long grain cross, we don't really even need to focus on size and shape. It doesn't really segregate. A lot of the amylase, it doesn't really segregate. So if you stay within the class, now you can focus most of your selection on the milling and the yield. So I just raised that point because there's a lot of great things about all this diversity that's out there, but you bring in something that has good diversity for a trait, a disease, or whatever, it often comes with bad alleles for all these other things. So if you take one step forward, you might be taking 3 or 4 steps back. So obviously, yield and quality are not the only traits that are important. So there are many other traits that impact the quality and the yield, and then there's also many traits that impact the profitability for the grower. So one huge one in southwest Louisiana is this herbicide resistance. The main weed we deal with is a wild red rice. So it's a real big challenge because all the herbicides that work on rice or on the red rice also impact our crop. So you're really limited on that. There is that clear field, but as you can imagine, we talk about weeds evolving to Roundup through mutation. You can imagine how quickly this can evolve when it just outcrosses. So the stewardship programs, you have to be very, very stern on those. But we do have fields where there is weed-resistant red rice. And actually there's a new system coming out. It's an alternate mode of action that will help those fields. But anyway, it's a complicated thing. So herbicide resistance is a almost must-have for this region. Diseases, there's probably a dozen more than I could even list that will affect us, but they're sporadic, they're not regular. We have 4 diseases that we screen with. We have a pathologist at the station, Dr. Don Groth. So he'll do all of these screenings for these 4 major diseases, insects, and then the agronomic traits, mainly standability and also that retaining capacity. All right, so here are some of the considerations, I guess, I've thought about when thinking about how I want to structure my program. And so first is the clear prioritization of the breeding targets. So what do I really need to deliver that's going to make an impact to the growers? What are the strengths and the weaknesses of the starting germplasm? So in this case, I said the breeders have done a great job on a lot of those quality traits. So, well, that's great. So I want to be careful not to disrupt the progress they've made. What are the resources required for each of these breeding activities? So I commented the milling is a big bottleneck for us. What are some of the logistical constraints? And this is the one that kind of always comes back. You have an idea, and you discuss it with some people, you think about it, and you realize logistically that's not going to work as far as in time or in labor. What are our target environments, and what are the key sources of variation in our testing program? So how many locations do I need? At what point is it overkill? If I have a finite amount of locations, which ones are the most informative? Which traits experience a lot of G by E? Those types of questions. And then finally, there's a lot of technology and shiny things out there, and then kind of always coming back and saying, okay, once they focus on what my end product is, worry more about getting to my goal and not so much the path I took to get there. So one way to think about these traits is basically just 2 things I think that really play a major impact in the progress you can make in a trait or the way you go about breeding for that trait. One is the genetics. So is it simple or is it complex? And the other is how well can we phenotype it? So phenotyping matter, how accurate can I measure it? How fast can I do it? What's the throughput? Those types of things. So taking these 2 things together basically gives me an idea of the relative breeding difficulty or ease. So something like herbicide tolerance, very easy to phenotype, very cheap, high throughput, single gene. It's, very efficient to breed this trait. Something like weevils, that is a complex genetics. You have to dig these things up out of the ground. It's very, very difficult to phenotype. Very difficult trait to breed for. So along those lines, we can tell you, okay, is this going to be a short-term or a long-term breeding target? And also, what is the investment for you to gain going to be? So if I look at weevil, which I said is very difficult, to make genetic gain in that is going to cost me a lot more than in herbicide tolerance. So thinking about those things. So now I'll talk about just the kind of the core product development program or variety development program that we have established currently. So basically, as we progress, the number of entries goes down as we move along. So we start with over 100,000 single, say, F2 plants. We select on a single plant basis for highly heritable traits. And then we look at by rows, and the things that get advanced from here are what go into yield tests. And at each stage, we're essentially looking at a plant or a row or a plot, and even on phenotype today, predicting, okay, based on how it did this year in these locations, I'm predicting it's also going to do well next year. So the phenotypes are also just predictions because they're limited. This is one location, so 1 year, one location. So there's no guarantee that how it performs this year is how it's going to perform in the future. If it was, we would test 1 year and release. So anyway, just the idea that the phenotypes are also just predictions. So as we progress with less number of entries as we move along, also the cost goes up. So the investment we make per entry goes up as we progress through this process. So when we look at, this is how our program is structured today. This is one location at the station, one location, one location. So at these early stages, we're looking at a single plant in a single year or a single row at a single location. So there's really no point in trying to say, oh, I want to select for yield or some of these complex traits, because what I see here, the odds of that translating as I move along are slim to none. So we focus on the observational type traits, so basically things that are highly heritable and things that we can look at and say, okay, this plant is too tall, this plant is too late, the grain type is off. Then as we advance, here are where we get into the traits that we're really interested in. We start using multiple locations, replicated plot data, so now we can start getting a reasonable estimate of some of these quantitative traits like yield and quality. We start getting disease scores that are over multiple years. So with this, you could say, well, maybe you could just do more here, invest more money for the phenotyping. So logistically, this is one of those things that, okay, if I had all the money in the world, or if I wanted to reallocate more resources here, we still can't test for yield and quality because it's a single row. Even if you said it was accurate, we'd have no seed left for the next year, so that we don't have enough seed at these stages to do some of these traits. So it's not just a resource or a time thing. A lot of it is a seed issue. So when we look at how we're thinking that markers can maybe play an impact on this, so again, first we're looking at this as the core breeding stages and activities we're doing, that will stay the same. We have something that works. There's successful varieties coming out. So kind of how can we layer some of these new technologies into this? So in general, at the beginning stages, we see markers can play a pretty big impact. But as we progress, it's ultimately going to be field performance. So each step, maybe the weight of how much we put towards markers versus phenotype will shift. But by the end, it's 100% field-based, and how it performs in the field is whether it's going to determine whether it's a variety or not. So one of the ways we see markers fitting into this system and playing an impact is can we start some selection at this stage on, say, the yield and the quality are two main targets. So now we're waiting to hear, but when we get here, we only have 2,000 individuals. So can we make some selections here when we have larger numbers to increase our selection intensity for those traits? The other is to increase the accuracy of some traits. So some of these early stage things, for large effect genes, can we select on a marker for a disease trait or a large effect quality trait that might be cheaper, but it actually might be more accurate. So blast is a big disease for us. Some years we don't have blast. So the F2s that year, even if you could phenotype and get a good score for cheap, if blast isn't there, there's no selection. So in that case, you'd really be increasing the accuracy from zero selection to marker-based selection. And finally is reducing the cycle time. So a lot of times you'll hear, these markers are going to reduce the time to variety a little bit, I guess, but you still need to do these years of testing in the field, and the markers are never going to replace that. So it's hard to eliminate this step, and this is a bulk of the time in the variety development process. But we can increase the cycle time from how long it takes to get something into a yield trial, and also, very importantly, how long it takes to get something where you're ready to recycle it as a parent, and that's really going to drive your rate of being there. Okay, so what are some of the applied molecular breeding activities that we're thinking about and see fitting into this? The first is general marker assisted selection. This is great for those large effect traits. It does require the upfront investment to identify that locus, validate that locus, validate it in your germplasm, in your environments. But once you do all those steps, it's very efficient for these qualitative traits. One of the logistical issues with this is it requires very, very large population sizes. So if you want to end up with 100 individuals to actually test, you need to genotype a lot more to get down to that 100. So that is as an issue of how do you manage that many samples. And the other is the whole genome prediction, which most people are very familiar with here. The benefits of this is that we don't need any upfront investment. We don't need to know the specific genes. It's part of our regular breeding activity, so there's no extra phenotyping resources or things like that outside of what we routinely do. And this is very efficient for the quantitative traits. So these two things together complement each other very well as two different approaches to approach different traits with different strategies. So when we look back at this little figure with our traits, really the traits that we'll target for marker system selection would fall up here, and the prediction traits would fall down here. Now, I guess you could do predictions for all of these, but for the case of, let's say, blast versus single gene or herbicide tolerance, I don't need to genotype the entire genome to tell me that it has this one specific marker. So we'll try to fragment these things from a cost standpoint. So kind of walk through how we are initially thinking about structuring this, and we kind of go through these iterations and then go back to the logistics of does the timing work and does it fit into this core testing process that we already have in place. So new breeding process and each of these steps. So the first thing I'll just say here is we will do a fingerprint of things after they advance from this stage to this stage. One, because the numbers are now low enough, but most importantly is because it's from here to here is where we recycle things to make new breeding crosses. So that's where we want to have a decent coverage on the fingerprint. So we walk through the marker assisted selection would really be early stage F2, maybe F2, F3 for these qualitative traits. So the numbers that we've kind of forecasted that we're going to need to really implement this is somewhere around 40,000 to 50,000 samples a year. And we can spread that out over a year, but the numbers get very big very fast. But this will allow us to not reduce any of the numbers that we put the rest of the way through the pipeline from what we're currently doing. The other is that at these stages, we already phenotype for all of our key traits. They all go into different disease nurseries, the milling, the yield, all of those traits. So we're going to phenotype these just as a normal process in the breeding program, but we'll then use this information, and that will be our training set. So again, we're not doing any extra discovery outside of our normal breeding activities, but this information will feed into a training set. The good thing about this is every year it gets updated as the relevant germplasm that's coming through. It gets updated in different allele frequencies, but also the environments. If we have a year with a lot of sheath blight versus none, it'll capture that. So we'll use that information. That will be what we use to make our predictions. And we see two ways, at least initially, of utilizing these predictions as part of this pipeline. The first is to start shifting some selection to the earlier stages in this pipeline. So like I said, normally we're selecting on, our first yield is on one location, 2 reps for milling and yield at this stage. With 2,000 entries. Can we initiate some selection at an earlier stage to make that basically the funnel larger? So in this case, for the progeny rows, we usually grow out somewhere around 50,000 progeny rows, and today we're doing that phenotypic OBS selection. Is it too tall? Is it too late? Is it covered in disease? So we still will do that because there's no point in genotyping this material only to find out it's too tall. So we can see that easily. So our strategy with this is we'll still do, as we always have, the OBS selections. We might look 40,000 rows, pick those that could potentially be products for those high heritability traits. And then we're in the area of 7,500 or whatever, 96 goes into that. I think it's 7,600 or something. Rows predicted a year, so we go from 40,000 to 7,000 based on phenotype. And then how we go from this 7,500 to to 2,000 would be based on the prediction. So what we're hopeful that these predictions will basically enrich the quality of yield and yield and quality that enters into that PY. We're not changing the number of entries we put into that PY. It stays the same. We're just initiating some selection a bit earlier. The other is we still can utilize some of these predictions even in the stages that we test on. So primarily, especially this preliminary yield test, again, we'll have phenotype data, but it's 1 year, it's one loc. So we want to start thinking about how do we weigh that? Do we count 60% based on phenotype, 40% based on predictions, or 80-20, or what? I think in reality, we're trying to find some collaborators. We're starting some work with Susan and Jen here to look at these things. But it'll probably vary by each trait. So in a year where we have 0 disease pressure, well, it'll be 100% for that disease based on markers. In a year where we have very good disease pressure, maybe we'll put more weight behind the phenotype. So trying to figure those things out. And then the last thing I don't have on here is, maybe I do. No, I don't. Is population prediction. I think there's a lot of interest in exploring that down the road of can we predict which populations to make, and also how big should those populations be based on what the projected variance around a given trait for a population. All right, so the real challenge, this slide is actually pretty easy to make, but the real challenge is how do we fit all this stuff in on top of the normal activities that we're doing here. It's there's a lot of activity we're doing in the field. We have locations around the state. So how do we incorporate all this stuff without disrupting our normal everyday breeding pipeline? So we have a few different pipelines. I won't go through all the detail on this, but basically it's not feasible to send everything through one approach. We need to spread it out across the year. Also, not all populations need to get managed the same way. So for example, a lot of these crosses aren't even going to segregate for some of these large effect traits because we've already done a good job in breeding them. So there's no point in doing any kind of marker-assisted selection. So basically, we estimate, looking at our pedigrees and the material we're making a lot of crosses with, probably about half of our crosses would be suitable or reasonable to do marker-assisted selection. It's segregating for a major blast gene or a quality trait. And the other half we won't do marker-assisted selection. There's nothing of a large enough effect or a big enough economic relevance that we would make that investment. Another thing is we have a very nice winter nursery in Puerto Rico, but there's a finite number of rows. So if we go through the winter nursery, we can test just in the second year, which is very nice. And it goes, again, to turn cycles over faster. But unfortunately, we just can't logistically handle everything there. So we have some populations that would get tested their first yield test in the third year, and then some in the second year. And that would be prioritized based on how important is that population in how important are, what are you trying to get out, what's the gap for the growers as far as varieties. So with this, we have these things spread out, and basically this is marker-assisted selection activities here in blue. We've structured these in a way where we have a really big push for marker-assisted selection in February, where it's kind of slow for us. We start planting in March. So we have a lot of hands who can help with all that sampling. Then we have a big push in July or June. That's the busy time in the field. So really we can't be involved in that at all. But the good thing about that is the associates who run the lab, we have unlimited student help. There's students available. Students aren't available in February, but the staff in the station can help. And then the last big push is over November, December, January, February. Again, it's a pretty slow time at the station, so there's a lot of hands on deck. So we think, we're estimating somewhere in the vicinity of 45,000 samples, 8,000 lines for whole genome genotyping. So we're up over 50,000 samples. So a big challenge with this is figuring out how are we going to structure this where we can keep all of our everyday activities going on, but yet collect that many samples, extract that much DNA, and keep everything on track. Okay, so I want to talk a little bit, so everything I've talked about now is pretty applied. We still are very applied program, and it will remain that way. But what are some of the research questions and things that we'll have? A lot of this stuff we hope to find collaborators where we could kind of complement each other on this, and then some of the things we'll do at the station with graduate students and so forth. So the first is, again, this is our kind of core activity, our elite breeding pipeline. Well, they shouldn't have all come up at once. But anyway, thinking about germplasm, the top here is about germplasm, and thinking a lot about how do decisions I make about what crosses I make and what germplasm I bring in will affect things like how big do my populations need to be, how many markers do I need, how many traits do I actually need to select on. So we do have a great source of new elite germplasm to come in. We do a shared nursery across all the southern rice states where we all submit a few dozen of our elite lines, and everyone grows them. We share that data, and we're free to breed with those. So that is a great source for us, because if the line is elite in Arkansas or Texas, it's going to have all the same criteria for us. So we'd be much more comfortable bringing that in, but they often bring unique genetics. The other piece is this diversity in germplasm, and this is where we need to be really thoughtful about what we do. The first question, how much of a gap in my elite space is there for this trait? So, so one, if a new disease arises, we don't have a BLB is a big disease in Asia. If that comes to the US and is as devastating here, well, we're going to have to bring something in, and it might bring some bad things with it, but you just need it. But for other traits, weighing what are the pros and cons of bringing this in. The other is the architecture of the trait. If it's a single gene, it's very easy to bring it in without disrupting things. We can back cross it, markers, and you can clean up all those other traits. But when they're quantitative traits and we actually have to try to pre-breed them in or cross them in, kind of thinking about how much effort is this going to take to do this, but also what are some of the thresholds. So am I willing to sacrifice a little on grain quality to get this trait in? And that will vary again. If it's to bring BLB in, which is just, let's say, killing all the rice, you're going to be a lot more flexible to say, okay, I'll take a hit on grain quality because I need this. But if it's something else, say straight head resistance, which is a physiological disorder, say, okay, it's great, it has straight head resistance, but I'd be much more strict on how the quality of that, the grain quality before I bring it into this space. The other is around trait and marker discovery. We don't do too much discovery on our own. The public in rice is gold mine. I mean, there's so much information out there, new papers coming out all the time. So a lot of our focus is on, let's take a look at what's out there and see what would work with us. Is more around the validation, identifying the markers, making sure it works in our germplasm. Some of the questions though we ask is, what's the size of the locus, and even if I had a marker for that and I had it in my germplasm, would I allocate the resources to do the marker assisted selection for it? So asking these questions to ourselves first before we make a large time investment to do all this work, but I'm not going to actually use it in my applied marker assisted selection. We probably won't spend much time on the discovery piece. And then the last is kind of an ongoing activity, is just really trying to optimize the number of locations, how we phenotype, and just how many reps, how many measurements, where's the trade-off? At what point is it too much and you don't get any more information from those measurements? These early stages, one location, this is always going to be on the station. We can't logistically do it anywhere else. But at this stage, we're talking 3 locations. Up here, we're doing 9 locations. So of these 9 locations, which would be the best 3 for this stage? So we have really great data sets over the last 20 years to look at this stuff and try to fine-tune kind of how we allocate our resources within this space. And then there's a lot in the prediction space, and really we're looking for collaborators to help us in that area. So that's models, what's the optimal marker number, accuracy, what are the trade-offs in that space. And then finally, developing some indexes for selection. What's the best ratio of phenotype to prediction at these different stages for different traits and different years, and ultimately hoping to automate some of these activities. So a lot of this stuff kind of a 5-year goal. We hope to have a lot of this stuff answered in the next few years. So one of the enabling things that is allowing us to do these samples is the genotyping. We just purchased this whole system from LGC. It's called they call it the SNP line. It's based on their task marker chemistry. Basically, it's for SNPs, small indels, and the great thing about this is it's very low cost per data point, and it's very effective when we have high samples. So like those marker-assisted selection things, talking 40-some thousand samples to run 1 or 2 or 3 markers on. So this system is very flexible for that. It's good up to a medium density marker, a few hundred markers, you can use this. And it's high throughput, so we can, in a day, do upwards of 8,000 data points. So everything I outlined today, this system has enough throughput to allow us to do this. And we have two wonderful associates who run this, so 2 people basically. And at any given time, 1 person can run the whole system. So we've been really happy with this. We just got it up about a month ago, and it's pretty much working right out of the box. One of the things we're spending a lot of time on with this is DNA extractions. So if you have a 50-cent DNA extraction and you're only going to run 1 or 2 markers on it, that really, 40-some thousand samples, that'd be $20-some thousand in DNA to run 1 or 2 markers. So we've spent the last month playing with some kind of cheap and dirty DNA extractions, getting them to work with this system, and we found that they do work. We've got that to work. So our cost for a lot of these things, our sampling, we do that directly into a 96-well PCR plate, and then we extract in that plate, and that's our stock DNA plate. So the biggest bottleneck in this whole thing is collecting the leaf sample, and if anyone's got any good ideas, I haven't found anything out there. So we're just doing it with tweezers. We do it in the greenhouse. It's almost impossible to do these numbers in a rice paddy. But we can do about a plate in a half hour. The extraction is very quick. It's basically you add a buffer and incubate it for 5 minutes and add another buffer. So that is not an issue. The cost for our DNA extraction with all consumables is under 7 cents, so that's very good. And then the genotyping, we do it in 3D4 or 1536 plates. If you're using the 1536, it's much more cost effective. And these costs reflect everything, all the consumables, and we haven't tried to scale these costs down yet, diluting it or trying smaller volumes. We're just kind of doing it out of the box, but less than 5 cents, and that's all consumables, tips. It doesn't count time though. Okay. So our sampling, this is an issue when we started. Wow, how are we going to keep track of all these things? How are we going to collect all these things? So what we found are these trays. This tray is 1 foot by 2 foot, and it's in a 96 well format. So this tray, these are 4 96 well plates essentially. So the way we're going to track things, just basically be from tray 7, 7A, 7B, 7D, and is it an A1 or H1 or so forth. So we can track these samples pretty easily. It doesn't take up much space. As we outgrow our space, you can stack these things. They don't stay in here for too long. So the leaf goes directly into a PCR plate. This is used for the extraction. And then these 4 plates are merged into a 384 plate, and that's our working stock. So there's a perfect relation to any flat in the greenhouse that corresponds with a PCR, a 384 PCR plate in the lab. So we don't have to worry about tracking single plants or anything like that. We know from this well, okay, it came from this well here. So basically all the tracking is just, okay, we want, from the marker system selection, I want these 50 plants. Okay, this one is in A1 from plate A, C10 from plate B, and we don't need to track them by one. Another thing is the planting. So we just bought this off the shelf vacuum, and basically spread the seeds on there, hold it, put it over here, and it deposits the seeds. It's not perfect. It works a lot better for the medium grains than the long grains, but it'll get you 75–80% of the way there. So you can plant one of these flats in probably 3 or 4 minutes. Because after you do this, you'll see, I don't know, not every little hole has a seed. Some have doubles, and then someone needs to go through. But when we were doing it by hand, it took 10–15 minutes. So that's what I'm saying. Every time we do something, oh, it's a great idea, we'll track these things, and then you do it. So, oh, it's going to take a whole day to do what we want to do. So luckily, there is a lot of stuff out there if you just look around, and this is a simple thing. We bought this off the shelf, and we probably could make it pretty easily as well. So this is the planting. These are the plants. We try to stay on a 7-day schedule, so we would plant. 7 days later, the plants look like this. We would sample, and then we hope to get the data back, analyzed, and the selection is made within 7 days. So after 14 days, we'll go in and just pluck the plants we want to. We don't kind of keep track of them. It's just enriching out of these. We'll pluck the plants we'd like to keep. We'll transplant them into, I don't have a picture of it, but these very small tubes. And this tubes can hold 102 square feet. And again, the idea with those tubes, try to go as small as possible to give us one reliable panicle. And that's what we need. We need one panicle from this stuff because it would go out into the field into a panicle row. So logistically, so far, everything is coming together. I guess we'll see this year and the coming years. I'm sure it'll look a bit different as we move forward. But so far, I think we have a blueprint that we think is going to work and we're going to move forward with. Okay, I'll summarize now, just about out of time. One, a lot of our breeding priorities are just determined by the market demand. So how many of our growers are affected by the trait? How much of an economic impact does that trait have? And that sets what our breeding priorities are for Louisiana. The demand for the grain quality, it's always been there, but it just continues to rise. People are demanding better rice. Last week there was a speaker at this conference we had in Texas who was saying that in the global market, rice is starting to become, just rice itself is starting to become more of a product than a commodity. So there's a really strong demand for this. We certainly need to continue or even increase our breeding focus on that, but at the same time, we need to maintain our focus on the yield. So some of these technologies, I think we're hopeful that will allow us to provide, or put more emphasis on the quality, but not sacrifice too much on the gains we're getting in yield. It's really critical, at least in our situation, to manage the genetic diversity, both maintaining what we have, so we don't want to, bottleneck what's already in that elite space, but at the same time be very thoughtful about what comes in into that elite space. These new technologies, they're all over the place. They're coming out all the time. Can't even keep up with them. Offer a lot of exciting opportunities. I think we're at this stage now where a lot of this is becoming cost effective to actually implement in a public breeding program. But we really need to be mindful that you can't slow down. You can't say, oh, well, I'll do all this, but then I'll make my crosses a year later. You can't disrupt the normal breeding cycle while integrating these things. And then finally, just a comment. The foundation of the breeding program is really going to be built on germplasm, your testing, how well you can collect data, manage your plots, and then a strong logistical infrastructure, managing your seeds, all those types of things. Really, all those things should really be well in place before going on to the next step. I guess that's just my opinion. I'm very fortunate and excited to be stepping into a program where all these things are in place. We have great staff, all the equipment, great data, managed seeds, and so this is a logical next step. But I would just say, if I was stepping into a brand new program or opening a program in a state that didn't have one, a lot of these things I talked about today, I'd probably put on the back burner, know what's on the horizon, but focus more on getting good plot data, getting the appropriate equipment, getting good staff, managing my seeds, and so forth. So with that, I'd like to acknowledge a lot of the other project leads at the faculty at the research station. Ms. Mona and Ms. Jennifer are the two associates who run the genotyping lab up for a year. These two women have run our DH lab for many years. No experience in the whole genotyping space, and they've done a wonderful job, and they're pretty much running that on their own already. And then within our breeding program, and there's one other person I should mention, Karen, but we have wonderful staff. I've learned so much from them in the last year, and will continue to rely heavily on them. But very fortunate to have a great established team already at the station. The other thing I'll mention here, we're very, very well supported by the Louisiana rice growers. Almost all of our research at the station, in one way or another, is either directly or largely supported by the Louisiana rice growers, which is also why I go back to our focus is for these Louisiana rice growers. We have a checkoff fund in Louisiana, so when you bring your rice to the mill, you pay a checkoff or a fee that goes back. Some of that money comes to us for research, and then some of that money goes to a promotion board. And rice in the U.S. has a very strong lobby. So we work directly with and for the growers, and we're very thankful to have that continued support. And having support like that really allows us to kind of plan for the future. It's there. We're very confident it will continue to be there, so we can start making investments today with the assumption that that money will continue to be there as we go forward. So with that, I'll stop and just say, if you're ever in the area or interested to come down, we have our field day. That's at the end of June 29th this year. It's a pretty big event. You can see all the stuff. We get usually about 400 people come out. And also, if you're interested, we release, LSU is very good. They release a lot of brochures and management tips for all sorts of things. This is our rice varieties and management tips, and you can find this if you just go on the LSU Ag Center website and type in rice varieties management tips. But we update this every year, and these are the varieties that we recommend to the growers. And there's economic breakdowns, breakpoints for inputs, costs, seed, all that. And, it's a nice thing. So with that, I'll stop and be happy to take any questions.
You discussed briefly the cooperative nurseries across states. Mm-hmm. So I assume those have been ongoing for years.
Many years.
Yeah. Is there any thinking going into how they might change as a result of marker technologies, or you want to just stay the same, like the way they are?
Change in what sense?
Well, this is sort of a soundbite, but the idea that you replicate alleles as opposed to lines, right? And my experience with these nurseries is that everybody submits a half a dozen or whatever lines, and they get highly replicated all over the place. But what about this idea of everybody submitting many more lines? Each line would only get planted once in Texas, once in Arkansas, once in Louisiana. I don't know, something along those lines. I mean, I'm not sure if it's a good idea, but whether there's discussion.
There's no discussion of that, and a lot of that it's called Uniform Rice Regional Nursery. It's 200 entries. 80 of those entries are rep 3 times. Those are like the advanced, and then 120 are rep twice. So it comes out to 480 plots. Maybe doing something like what you're saying as an additional kind of thing, but I don't think it would replace this, because this is our way to evaluate how a variety a few years before it's released from Arkansas does in Louisiana. It would help whether or not we would recommend that. The other thing is, this is our way to see experimental lines coming up through Arkansas or Texas or Mississippi or Missouri, and decide, do I want to cross with that? So I think everyone would want to be able to eat the whole collection in their environment. But you're talking maybe doing something to help make an estimation set or something.
Yeah.
Yeah. Right. No, I think that would be I think very useful. It would just probably be something maybe we would add on in addition. Yeah.
You mentioned that the importance of looking at quality characteristics to meet export demand, but you didn't mention in your slide on quality, you didn't mention nutritional quality. Do you think there's going to be an export market for, let's say, biofortified rice that contains better nutrients?
Yeah, potentially. We haven't had any feedback from the millers or the exporters whether they've had much of a demand for that. But absolutely, if that word came back from them that, hey, there's a sizable market, people are demanding this, then that certainly would go onto the list. But
I was kind of interested in, you were talking about early genotyping for just marker-assisted selections, very low coverage, wider range, and then later you might do some whole genome. Are you, like, logistically, are you extracting DNA twice on the same lines, or you just extract the DNA already, so you just, and how does the price kind of balance out? Obviously it's much cheaper per individual earlier, but you are doing kind of double genotyping.
right.
We're really
Yeah.
simplistic if we're just doing whole genome multiple times.
So the early stages, for a single marker, 2 loci or something, that would get extracted, I guess we could say an F2. Once those plants are transplanted, all that DNA would just get thrown away. We would need a warehouse to keep all of it. So we run that. Once those plants are transplanted, all those plates get thrown away. Those F2s would then go to be, I at least F3s. And let's say in that case, F3s would be the rows. We would go observe those rows, and the ones that we wanted, let's say we observed 40,000 F3 rows, and we picked 8,000 rows that we were interested in trying to predict. We would take that F3 seed, harvest it. Some of that would be stored if we ultimately decided to test it, and we might just germinate, one or two of those F4 seeds, genotype it, and use that for the predictions. So essentially, I guess you would be extracting it twice, but it would be a different generation, and we just logistically it'd be too hard to hold all those plates.
You showed a 7 cent per marker. Was that a per data point, or is that if you wanted to do 5 markers, would it be 35 cents or?
Well, so that cost of 7 cents is the extraction.
Oh.
So let's say 7 cents for an extraction.
All right.
After that, the cost per data point is let's just say 5 cents, a little less than that. So if you were going to run 1 marker on 1 individual, the total cost to you would be 12 cents. If you ran 2, you already did that cost for the DNA, it would be 17 and so forth.
I have a question about U.S. demand for rice, and I'm wondering what sort of rice we import into the U.S. primarily, and whether those are kinds of rice that can be grown in the southern states.
Yeah, I think a lot, and don't quote me on this, but a lot of what we bring in are the specialty rices, the basmatis, the Thai jasmine. A lot of, I think, what people eat, kind of go back to the previous speaker, where they're from. If you have a population of people somewhere in the United States that are from one of these countries and they have their special rice or their Italian rice and all that kind of stuff. But most of the rice that's consumed in the U.S. is produced here. When the export market for some of these specialty rices, what I understand, at least what I've heard, is when LSU first came out with a jasmine, so there's a Thai jasmine, and they called it Jazzman, like jazz music. When they came out with that, it's very good rice, but on the export market, I think it grew to 40,000, 50,000 acres in Louisiana, but then the Thai rice exporters cut their price, undercut the price, and it knocked it back. Right now, it's a specialty thing. It's on 5,000 to 10,000 acres, and it's for specialty things, stuff you see in the grocery store where you pay a lot more for it than the big sacks you might buy. Juan?
You mentioned the opportunity in the season. Is there a genetic variation for the trait? Is that something that can be
Yeah.
exploited?
Yeah, there is genetic variation for it. So that is a trait, at least in South Louisiana and Texas, that could kill a variety. So there's not that many traits that will just kill a variety, but that's one. Obviously, yield and quality are others, but so it needs to have a good retooning capacity. Again, that's something that breeders have done a pretty good job on, but we've never done. There hasn't been too much work on trying to understand that. We do take on our yield trials. We do take a second. We do cut it twice.
Could you talk a little bit more about population prediction and why you might be using that?
Sure. I'm certainly not the best person to be talking about this, but the idea, my interest in it is we can basically simulate what a cross would look like and then predict those progeny. And I see value in that, one, to determine which crosses. I have a finite number of crosses I can make every year, and there's more crosses could be made than we can actually handle. So now we might use phenotype pedigree data to make that determination. This would just be another layer to kind of predict, even though all 4 of these lines are equally good for, let's say, all the traits, how would the combination of those alleles differ in this population? So one, it might say, well, actually, it's not that good, I won't make that cross, or this cross is better. But another thing I'm interested in exploring is how big should my population be? Like right now, it's kind of like every population is the same size, but based on predictions, we can get an idea of, hey, this population is really good, it's got a high mean, but the variance is small. Then I might want to go forward with that population, but I don't need to plant a lot of individuals. Where the flip side, you might have another population, if the predictions say, hey, the mean is really good, it's equal to that other one, but there's a large variance around it, I might want to plant more for that one to try to get further up in that distribution. At least that's my ideas, I'm certainly not an expert.
We have a question from the webinar. Has the recent flooding damaged your regional trials or commercial rice production fields? If so, how will you deal with the loss of data?
We haven't planted yet, so last week was we planted our first trial last week, and that's a date of planting thing, so it's just we're planting early. A lot of that flooding that's happened is actually in the northwest part of the state. When I flew out yesterday, when I went to bed 2 nights ago, I thought there's no way I'm getting out of here. We're supposed to get 10 inches of rain. And when I got up in the middle of the night to the airport, it hadn't rained. I talked to my wife yesterday, she said it never really rained. So most of that, what you're seeing on the national news is like northwest Louisiana. We don't really grow rice up there. And even if we did, we haven't planted yet. Some people started planting last week, but it's very early.
So you mentioned that herbicide is very important, and right now we have the clear field. But to what extent are you doing exploration for novel sources of herbicide resistance to other herbicides, so that
Sure.
this is over?
That's a very active research area. So BASF does a lot of work in that area. We have a biotech program at the station, which is essentially mutational breeding for herbicide resistance. A lot of the other universities do as well. So there's really no GMO work in rice. They had an issue with that 10 or so years ago, and the market does not want that. So BASF has a new mode of action out called Previsia. We were given that germplasm first rice to breed with that, and we hope to have a variety out next year. And that is an alternate mode of action. It's not as good as the clear field as far as the herbicide. It's not as broad spectrum. There's no residual. But if you have clear field resistant red rice in your field, this is a good option to work into that rotation. So there's another one coming out. The hope with that is, well, one, you can clear up your red rice fields, your clear field red rice. The other is it'll help doing those in rotation. So one of the weed scientists at the station, Dr. Eric Webster, he's doing a lot of work to figure out what's the best rotation to do. You do Roundup beans, clear field, Previsia. I'm just trying to figure out what's the best to sustain all of these technologies as long as possible.
One last question.
Could you maybe provide a little bit of insight into the market dynamics in the U.S. of quality between hybrid and open pollinated varieties? Because I know in some of the Asian countries where hybrid rice is trying to take off, there's issues with quality and the consumers don't like it. But in the U.S., most people are not nearly as discerning about rice. Buy rice because it's cheap. They don't really care besides that.
Yeah, that's a very good question. So the hybrids yield 20%. I mean, it's pretty much, they yield a lot more. There's a lot of issues with them. So hybrids aren't going anywhere, but at the same time, varieties aren't going anywhere for a long time. So the hybrids have that yield advantage, no doubt about it, but the quality is a huge issue. Also the height, the lodging. So there's a lot of those things that need to get straightened out in the hybrids, and I think it has actually harmed the U.S. rice export market, because we used to be like the gold standard in quality, and as more and more of this hybrid came in, we're 25% in Louisiana, Arkansas is 50%. Arkansas produces 3 times as much rice as Louisiana. So a lot of what is getting exported is hybrid, and the quality is a lot lower, and that's a few reasons. One, that uniformity I talked about. The hybrid, the hybrid itself is an F1, but the seed that goes to the mill is an F2, and a lot of these traits are controlled by the genetics of the seed. So within a lot of seed, a variety, the differences in the size and the shape and the moisture and all that kind of stuff are much, much more constant than in the hybrid. So that uniformity affects a lot of things. It also impacts the milling, because you have different sizes, so you get more breakage. Also, chalk is a big issue, that appearance thing. So the challenging thing for the hybrids is you could have 2 inbreds that look really good, but when you combine them, if there's different genetics make them look really good, and you combine them, now those things are going to segregate in the seed that goes there. So we have a hybrid program at the station run by Jim Ward, and his main focus now is to really just, the yield is there, but to try to improve those quality parameters. RiceTech is the main company in the U.S., and they're doing a lot of great work and putting some serious effort to improve that. And they have. And the hybrids they had 10 years ago versus today are orders of magnitude better in the quality. But I think what ultimately is going to happen is, again, it's a trade-off for any of it, but the people breeding hybrids are probably going to have to focus less on yield for a while and focus more on these quality traits, and then once they get those, go back to the yield. But the yield is already there. So I think as a varietal breeder, I'm not concerned. The varieties aren't going anywhere for a long time. And the hybrids, I have, the yield potential is there. So I mean, there's definitely a place for them as well. Okay, thank you.
Alright, hi, my name is Chris, and it's my pleasure to introduce today our next seminar speaker, Dr. Rich Ozminkowski, from the KraftHeinz Food Company. Rich came today from California, but he's actually originally from the New York area. He's a New York native, grew up in Rochester. He studied at plant breeding and horticulture at North Carolina State, and after that he went on to receive his PhD from The Ohio State University. After which he joined Heinz, where he's worked towards developing regionally adapted processing tomato varieties. And since 1999, he has led Heinz's global tomato variety development program, and has had the opportunity through that to travel worldwide to evaluate tomato varieties. So today he's going to be talking to us about variety needs of globalized tomato processing industry. So please join me in welcoming Rich.
I'm very impressed with Adam's talk. I think he did extremely well, and I'm glad I didn't decide to talk about our processes because it would seem like a lot of repetition. I'm talking tomatoes, he's talking rice, I'm talking hybrid crop, he's talking OP crop, but still a lot of the concepts, the resource management, it's a lot of the same thing. So, well done, Chris. I think maybe your time at Pioneer helped with that focus. I guess, I don't think I'll follow his slides. So do I just close that one? Say hi. As a corporate breeder, corporate scientist, we're notorious for not being able to say anything and talk about what we do. Hopefully, I'll at least be able to put some information out there that is meaningful. And certainly there'll be time for questions and answers afterwards. One component, though, to keep in mind, I am what is standing between you and lunch. So, thank you to the organizers, to Margaret, Anna, for Chris for the introduction. I really appreciate the thought, and it looks like it's a great opportunity to talk to a whole bunch of students and such to explain, you know, kind of the industry perspective, what an industry breeding program is looking at. And I think what we're in a unique position is because when you think of Heinz or Kraft, or as now we're known as KraftHeinz, you don't think of seed companies or a seed company. You think of food. And so what I'd like to do is take a little bit more of the quality aspects that Adam was talking about, and kind of build it even further. I'd also like to thank Pioneer for sponsoring the event. I think it's a great job exposing students and I guess the rest of the world to a lot of these topics. Oh, new buttons. All right. One thing I do want to make clear as well is I'm speaking specifically about processing tomatoes. While many of you are working with small grains and they're like tomatoes or tomatoes or tomatoes, the breeding, the quality parameters, everything that's looked at in most developed countries are completely distinct between a processing tomato and a fresh market tomato. You can certainly still put a processing tomato on your salad, there's nothing wrong with it. It's just what the market demands and the process of the harvest and what's required of it is completely different. All right. When you think of the KraftHeinz company, or maybe you just think of them as separate, we just merged this past June. These are the brands. There we go. These are the brands that you think of. You think of things like Ore-Ida, Oscar Mayer. You probably have a lot of these in your refrigerator right now. Think of it's a global food company. You've got, this is Indonesia, Australia, Brazil, Italy. This is Northern Europe. It's Poland. But like I said, we also have a couple of breeding programs. A major program that I'm going to talk about is the Heinz Seed tomato seed program. But we also do, and I just found this out recently, a small breeding program in California for Cornell. Okay. I hope that breeder's not online. So I'm just learning about that one myself. So just a quick little plug for the company. I'm going to try not to make this too much of a shameless commerce, but we were created just last year by a merger between Kraft and H.J. Heinz, orchestrated. Warren Buffett is one of our owners, 3G Capital. I can go on that later over lunch if anybody's interested. It's a $28 billion food company and is the 5th largest food company in the world. We have 8 brands that are valued at over $1 billion each. So, and we have a tomato breeding program. We're not valued in one of those 8, I can tell you that. But they do watch us. They watch us very closely on the accounting side. Okay. So we're a global food company. So for us, resourcing ingredients, resourcing supplies is a global issue. We have a single global procurement. It's, a lot of that is based out of the Netherlands. So we have to look at things globally. And I think as the world is becoming smaller, in terms of just communication, in terms of the rise of the middle class, we have to think of the food supply as global. All right, the growth of the middle class. I thought I'd focus on the actual topic by inserting information that's relevant to the growing middle class. I found this slide from a presentation on the Brookings Institute done a few years ago. This is the rise of the middle class, and I think this is the topic of our conversation today, and you can see how that's grown over the past 15 years. I guess this would be 2011, so at the time this is actual data versus projected. If you look at where we are now and where we're going to be going, you're looking at about 50% or more of the world's population is going to be middle class. Okay? The rich is not growing quite as well or as much, and certainly that's at the loss of the poor, which is a good thing economically. The other thing that they noted is there's a distinct movement in what's called the economic center of gravity to the south and towards the east. What it means is around 1964, it would be, say, if you took a map and put whatever economic factors that went into this, it would have been centered around Portugal. It's moved since 1964 until I think it's somewhere around, somewhere in the Middle East. So it's distinctly moved. And I don't think any of us are surprised at that as we hear the development in China, in India, etc. So the world, the economic center of gravity is moving in that direction. So as the middle class is growing in these developing regions, so does the demand for processed food and food away from home. I know the big trend in the United States is eat local, eat fresh, minimally processed, and that's all well if you can afford that, if you can access that. But as we heard from our first speaker, some of that stuff isn't even available. And so as other areas of the world develop, they want things that are a more Western diet. They may be adding more meat. Okay? They're adding more processed food as well. They want things like pizza. All right? They want ketchup. They want those sort of things. When you go to, as Burger King or McDonald's or whatever, Pizza Hut expand into those developing areas, you're getting a higher demand for, in particular, tomato paste, processed tomato products. And so over the coming decades, it's suggested that this demand is going to be explosive to meet that higher middle class. So I want you to think about, as we go through this talk today, is that it is a global food supply. It's a global processed tomato supply. So I wanted to go a little bit into some, actually I'm not an economist, so it gets into plant breeding a little bit later. When you look at the consumption, and this is in metric tons of raw tomato fruit, okay? Globally, in 2015 and 2014, we were just around the 40 million metric ton consumption in the world. In 1997, it was around 20 million. Population growth, quite significant over that time, of course. Increases in production or in per capita consumption, quite a bit of an increase. That is what's driving the demand. Because if you take this apart, the population growth only accounts for about 1.2% annual growth rate of consumption. And in reality, we've been seeing a compounded growth rate, annualized growth rate of about 3.3%. So it's the increase in per capita consumption that's driving the need for more tomatoes. And so we need to be able to produce that increased, or supply that increased demand with high quality tomato products, tomato sauces, tomatoes in general, that are produced, I'll use the term sustainable, and I'll get a little bit more into that. Okay. As I said, a global food supply by 2050, we're looking at 9 billion people. There was a great article, I don't know if any of you had looked at this. This came in last year, around May. The National Geographic did a series on food. And they talk about this, we're going to be adding 35% more people. To meet the global food demand, we're going to have to add, I forgot how many this is, about 10 billion tons of food to be produced to feed those people. And we're already using about 38% of the world's ice-free land. Now, there's certainly something, if you want to, we can breed for crops that can grow under ice, then certainly that adds our land that's available. But we've got what we can do, the land that we've got. And not to mention, you've got to house that extra population. Okay? We've got to be able to grow more food on the farms that we have. We need better varieties is a big part of it, as they indicate. Foley also indicates we need to use our resources more efficiently. We need to shift our diets away from livestock food production. So I'm glad Allison's not here to hear that. As you'll see later, that's an important part of her work. And we need to reduce waste. We need to reduce the loss, not just in the supermarket, not just in the refrigerator, but the losses between the farmer and the market. right? The disease that's taking down crops, the spoilage because of poor timing. Okay? Those things have to be addressed. We can grow the food. I think we can grow the food as long as we're not getting distracted by things that work against us, things that are going to reduce our production. We have to become more sustainable. That's what I'm using as a definition of sustainable. Okay? So this is what the American Public Health Association points out, as opposed to reading it directly, just to point out a couple of things. Healthy food, have to meet the food needs. You have to maintain healthy ecosystems. You have to have nutritious food that is accessible and affordable to all. Again, we're in the United States. We have a lot of luxury that we can spend a little extra on food, but when you look at a global, food supply, most people, or a lot of people, they're just looking to get the calories and to be fed. They don't have the luxury to worry about whether the beef was grass-fed or whether the chicken was petted as it was laying its eggs or something like that. So part of sustainability, too, is looking at the social, environment, and the economic components of the production system. So that involves good agricultural practices, responsible stewardship, producing more fruit per hectare, kind of a theme here I think maybe we're getting, producing more final product per ton, and meeting product quality standards. Not just producing the actual tomato, but actually producing more tomato paste, or producing more ketchup, or producing more pasta sauce on that acre of ground. And seed varieties, this is where it's coming around, seed varieties are critical in being able to accomplish that. So let me talk to you a little bit about the Heinz Seed Program itself. Henry Heinz, our founder with the nice mustache, he stated one of his many famous quotes. He was a marketing person. He was quite ingenious at that. How many people know 57, right? 57 varieties? There were never 57 varieties. That's marketing. So, sorry, now that's captured forever. I'll get in trouble from corporate, but I think to improve the quality of a food product on the shelf, you must first improve the quality of the produce in the ground. And I think that gets back to Adam's conversation. It gets back to what breeders have to do. We have to help produce a quality product. This is our guys put in doing some cultivation last year on our research farm. That was a joke. They don't let us use animals anymore. No. These are some slides from around the 1930s in here. So, we've come a long way. And another one. So the tomato breeding was initiated in 1936 in Ohio, northwest Ohio. And the objective, and this is a quote from our first report, which was the raw products department at the time, and we had to provide an abundant, dependable, and economical supply of high-quality raw products for our business. I'm going to ask you, in concept, isn't that sustainability? And that was 1936. I'm sure that had never been put on a billboard. The word sustainability had even been put on a billboard at that time, and most people wouldn't even know what it is. And that is how tomatoes used to come into the factory. You got the buggies all lined up. I wasn't there. So, where do we sit now as Heinz Seed? This is our research facility. We've got about 130 acres. This is the size of our research plot. We grow about 5,000 different lines of tomatoes, and this is our rotation space. There is no cover crop, I'll be blunt, because we have challenges with certain perennial weeds, and we find controlling them in a wheat crop to be quite challenging. We have our greenhouses, about 11,000 square feet of greenhouse space, and some of our laboratories. We are the global market leader in processing tomato seed, and it's all hybrid seed. We don't sell seed just for Heinz products. We sell it to anybody willing to pay the price for a bag of seed. We have sales in more than 30 countries, and we're the #1 sales or processing tomato seed company in these, among other companies. So we've got over 50 commercial varieties that are for sale. We only work on tomatoes that are intended for processing, and I'll build on why the word intended is in there in a little bit. We have a global breeding program focused in primarily, our center is in Stockton, California at this site. We also do Leamington, Ontario, and Victoria, Australia is where we have our winter nursery. We have 4 breeders. There's a total of about 35 people involved in the seed business and overall. And we do global adaptability trials. So, Heinz Seed has a conventional tomato breeding program. So it's T-harmony, right? That's what as a breeder. There's other words that they call breeders, but basically we use DNA markers. We don't use GMOs. Global tomato production. All right. Processed tomatoes, as I said, are a global supply. In California, last year we had about 14.4 million tons of tomatoes. These are all short tons. So about 14.4 million tons. Certainly that's the largest single production region in the world. North America, the rest of North America was about 1 million tons. That includes Ohio, Pennsylvania, a little bit in New Jersey, Indiana, Ontario, Canada. Europe, Italy has about almost 6 million tons. Spain, almost 3.5. Portugal, close to 2. And the rest of Western Europe at about 670,000 tons. We got Turkey is becoming a very important producer of tomatoes, of processed tomato products. Asia Pacific, a lot of this is naturally in China. Most of it is in China. Africa, Tunisia, and then South America like Brazil, Venezuela, and Oceania would be Australia, New Zealand. This is where the production has been. Tomato exports. There's a lot of exportation of tomato products. You look at, this is in raw material equivalent, so in metric tons. You've got about, in 1997 was not even 10 million, and now we're up at about 25 million equivalent raw tons of tomatoes, processed tomatoes being exported. And they're being exported primarily in tomato paste, as you can see, but also in some other products. Tomato paste is a very easy commodity to move from location to location. Okay. So, as our demand continues to increase for a rising middle class, as the population continues to grow, we have to look at being able to supply that demand. Now, do you supply it regionally? So if the demand is being increasing in India, is it being increased in Nigeria, which is an area that we've heard a little bit about? The thing is, you got to take a lot of factors into account. Tomatoes are not a subsistence crop. Okay? They're adding flavor to a meal. Okay? It's not something for just calories, like if you're growing rice, you're growing corn, you're growing cassava. Those are your basic calories. Tomatoes are a flavor. And they're not just calories. Tomatoes are perishable. Okay? Typically on a processing tomato, they're processed under 10 hours from the time they're harvested. So as you'll see in a few slides, they're harvested fully red ripe. Again, a completely different system, different qualities than a fresh market tomato. Very expensive infrastructure in order to do the processing. A lot of stainless steel, and you need a very highly coordinated harvest process. You can't just have people delivering tomatoes expecting to get paid on a per pound or per ton or basis. and whenever they want. You've got to keep the factories running. When a factory shuts down, it's an incredible cost. You have to keep the evaporators running 24/7. And on a global basis, you're fairly limited where you can effectively grow tomatoes. They're adapted best to a Mediterranean type climate. They don't like a lot of rain during the year. They like it dry. So the best places are places like California, Spain, parts of Italy, where it's mostly you add the water as it's needed. This is a photo from one of the largest tomato paste processing factories in the world. There is a lot of infrastructure here. This one factory, which obviously is not, the whole thing isn't captured, can generate paste from over 1 million tons of tomatoes per season. Okay, so there's tomato production being global. There are the sophisticated production areas. Places like California, Ontario, Spain, Portugal, Australia. As small as it is a very sophisticated system. Even to some extent, northern Italy. So what constitutes a sophisticated region? They have strong agronomic practices and they have large yields. Hundreds of acres in a field. You've got to have, for tomatoes to be grown most sustainably, most effectively, drip irrigation. Okay? Overhead is like rain. Tomatoes don't like to get their leaves wet. Promotes all kinds of diseases. Mechanization. You've got mechanized transplanting, because you're putting everything, those little plants go into the ground, and mechanized harvesting. You've got to have a very tight harvest schedule, and you've got to have it adhered to by the growers. And you have to have a strategic hybrid use. And this is what keeps production in these areas. These are areas, I mean, these are not known as low labor cost regions in the world. These are typically very expensive regions for the labor. It's a lot less labor goes into tomato processing than you might think. So what keeps these areas competitive is because they've dialed in on this. So the processing tomatoes in California, to go through the system briefly, you've got we do about 14.4 million tons last year. That'll be down a little bit this coming year on about 293,000 acres. 100% hybrid varieties. Growers can get anywhere from 40 to 70, and I've even heard of upwards of 90 tons, short tons, to the acre. Average last year was just short of 50 tons to the acre. And the price, typically between $70 and $80 per ton. Okay, not per pound, but per ton. So the crop is grown in fields. As you can see, this is a very large field. It's not photoshopped to get rid of things in the middle. And you sit and wait until the crop ripens completely, at which time you pull a harvester. Okay. The harvester undercuts the plant. The plant and the tomatoes go up into the harvester. It shakes off the tomatoes. The leaves go out the back. Okay. The tomatoes go around the belt. There's people watching to make sure you don't get rocks and snakes and all that sort of stuff in there. You don't want those in. And they do a good job. I should clarify, this is being recorded. There are a lot of stops before any of that stuff would get into the factory. Okay. And then the tomatoes go through the elevator. They're coming in over here, and they're dumped into these bins. One of these bins, this one is not nearly as full as it should be. This one tub would weigh about between 12 and 13 tons. Okay? So think of what those tomatoes have to go through just to get into the tub. Think of those poor little guys on the bottom. All right? So this is all, I bring this up because as you're breeding for a product, whether it's processing tomatoes, whether it's sweet corn, whatever, you have to think about the entire system. The tomatoes are dumped from these, and again, this is considered one load of tomatoes. That's about 25 tons. They're dumped into the flumes, washed in. This is where it's actually unloading. Okay? The tomatoes float, which is a good thing. The tomatoes, they float up. Here, the water, as you might imagine, water doesn't go up voluntarily. So they are being drained. And I know this is dirty water. This is California. Water is for, our whiskey is for drinking. Water is for fighting over. So there's a lot of water recycling in the process to try to minimize the wasted water. So you've got the tomatoes coming off. They're being sprayed down here before they go up into the later steps. They go through flumes. They go through again. There's flumes and flumes. This is the wastewater being drained off. Then the tomatoes, you can see, are on a white belt. And they're, again, working their way into the factory. So this is washing them through sequential series of water rinses. Okay. Again, they go through, they're evaporated. They're put into these aseptic sterile what are called Scholli bags, named after the company Scholli. So they are completely sterile and aseptic. One of these boxes, okay, they can stand out here for a couple of years and basically maintain quality. One of those is about effectively $1,000 worth of paste. There's about 1.5 tons of tomato paste in there. Each of those boxes. And that's probably somewhere around maybe 30 bricks, 28, 30 bricks. Tomatoes start at 5. So you've got about a 5 to 6-fold concentration. So to take that up, you've got, what's a 6? You'd be at, 1.5 times 6? You're at 9 tons of raw tomatoes would have potentially gone into that bin. These are the basic commodity that when we saw that big red bar about paste exports, this is primarily how it's shipped around the world. They fit nicely in railroad cars and the like. We've turned something perishable into a shelf-stable product. So, what are the challenges in these developing areas? I brought up areas like India and Nigeria, for example. Okay? In a lot of these areas, you've got hand harvesting. Okay? How are you going to harvest millions of tons of tomatoes if that's the demand? Or even hundreds of thousands of tons of tomatoes effectively like this to keep one of those factories? It's a challenge. You've got to get to mechanization. There's a very large grower base. Okay? Potentially in some countries, over 100,000 growers could be involved because they farm 1 to 2 hectare fields. Okay? How do you coordinate a harvesting process when you've got people that are just growing a couple of hectares of tomatoes? Unreliable supply, and this goes back to my word intended. While it's our intention, for example, to breed tomatoes that are for processing only, okay? In certain countries, the growers have to make a living. The growers have to subsist. So there's nothing wrong with this socially. It's more a matter of it's a logistic to be handled as you go to produce enough food. Is the growers are going to see, wait a minute, these tomatoes, I can get more money if I pick these tomatoes. Yeah, I was going to grow them for the company and go to processing, but I'm going to get more money if I do processing, if I do it in the fresh market. So suddenly what the factory was counting on coming into the factory, suddenly it's going off to the market. And that's a challenge when you're trying to have an efficient operation and keep it running on a 24/7 basis. Poor processing quality. A lot of places, growers are picking the varieties themselves. They want to keep that option between, can I take it to fresh market or dump it at processing? Okay? So when they're choosing varieties, they're chosen for characteristics for fresh market. Okay? Color can be horrible. As you might imagine, this is not going to give you very good pizza sauce or ketchup. The color is terrible. Also, the yield on those is not very good. When I talk yield, field yield, I'm talking specifically factory yield. A variety like this is going to give you very poor yield in the factory. So you don't have the quality characteristics. You need potentially novel pest resistance. For example, in certain areas you've got more problems with insects and such, which, as Adam mentioned, if you're dealing with weevils or such, they're a little bit harder to control than when you can just go out and spray with an inoculum for a pathogen. Seed importation was touched on this morning. That's a big problem, getting seed into countries, and sometimes it's used as a protection mechanism. Okay? Very poor variety trial systems. It's hard to run varieties when your growers are only 2 hectares in size. Your trial would exceed the size of their whole production. And they have often very poor agronomic practices that have to be advanced. All of these have to be sorted out before you get to varieties. I got this picture from in Argentina. You got a guy spraying a chemical, walking down the road, not just looking at his PPV, his protective, or PPE, personal protective equipment, is severely lacking, but look at all these kids and people are, this is not sustainable. So variety development. A variety is only sustainable within the context of a specific environment and a particular product. What may be sustainable in California is not necessarily sustainable in Ohio or in Australia. This is the crux of my slide, and I think it builds a lot on what Adam was talking about. Like I was saying, sustainability revolves around the product output per area. How many jars of pasta sauce? How many cans of peeled tomatoes? How many jars of ketchup per acre? Okay? And that involves 2 things. You have to have field yield. You have to have factory yield. And that's what growers say, which is the best yield. What's the best yielding tomato you've got? Well, what's the best factory yielding? What's the best variety for the factory? Well, it's too broad of a definition. Okay? So even that, you're looking at the best you can do is you say, well, you look at yield potential. Well, I've seen this do really well in other places. Adaptability, but I don't know how it performs in your place. I don't know how it performs to your cultural conditions for your actual farm's particular practices. And when you look at factory yield, it's the same thing. You've got to take it down a level. Fruit quality. Okay? I need higher fruit quality, better fruit quality. What's your best quality tomato? You need a predictable maturity window, harvest window. Is it early? Is it late? Is it mid-season? When does it come in? Even that, factory managers think they're giving you a lot more information, or growers a lot more information. But still, you can't breed and just say, oh, I'm breeding for, this is a high fruit quality tomato. You need still far more definition than that. So when you look at adaptability, yield potential is what it is, but you've got to look at the adaptability, which is a huge component. You need to look at disease resistances, field storage, so are those tomatoes going to rot in that climate? Reliability, so it's, you've got to have a stable growth. You've got to look at fruit quality. Okay, do you need high brix, which is sugars, just like grapes? You need to look at viscosity, which is important for things like ketchup and pasta sauce. You need it thick. You don't want runny ketchup. So for Heinz, viscosity is key. If you're not peeling tomatoes, why do you care if it's peelable? So a factory producing paste like this has really no concern about peelability. But if you're making peeled tomatoes, diced product, obviously this is a key to you. So you've got different customers with different factories, different product targets that are directing what their needs are. Color is typically valuable to most processed tomato products. Some more than others. You've got high-end output. You've got a company in Portugal that they look for particularly high lycopene tomatoes. That's how they're marketed. But even that is not necessarily enough information for a breeder. You need things like, okay, define peelability, wall thickness and firmness, shape and size, VHL, that's our trademark for very high lycopene varieties, reliable performance, you've got stability, but also you've got potentially water use efficiency. Disease resistance completely depends on the climate and the soil. So all of these things, and you could take this down. There was an earlier question about molecular markers. And I think maybe in a crop like corn, I can't imagine what other crop there might be that would have that, where you can have truly enough information that you think markers will replace observation. Because there are so many things that we don't know in those genomes, of those thousands upon thousands of genes, to truly think we can capture all of that in all the different environments. I don't think we're there yet. Can we get there? I'm sure. That's a lot of money, it's a lot of time to get there, and there's a lot of work to do it. That's a lot of opportunity for the students out here. But for me, I need to see how it grows in a half a dozen spots in Italy, or a half a dozen spots in Spain, or Portugal, or Ontario, 20 locations in California. Then I start getting a picture of the adaptability of that work. To me, that's what's important. So, breeding considerations. Tomatoes, like I said earlier, are primarily processed into paste. They're processed into diced and other products. And they can ship, even those can be shipped reasonably affordably around the world. If the product, for example, the Midwest, the big thing that keeps the Midwestern U.S. Tomato industry alive is not paste. The Midwestern United States cannot produce tomato paste, and certainly not Ohio, New York, to be truly competitive on the world stage. It's cheaper to bring paste in from California than it is to produce it here. What makes it valuable is because the demand for the diced products, for the peeled products, for the other things that are a lot less economic to make it into, or to ship across the United States. That is what keeps the industries alive. If all they needed was paste, it would be a big, it would be a whole different economic equation. Local production, again, must be competitive with major global production. Just because the demand is growing in one region doesn't mean it's effective to produce it locally to that region. And it must be sustained. And again, as I pointed out earlier, the variety isn't the only component. We can only do so much. And if you're looking at an area that's producing 1 to 2 hectares for a grower, they're getting 8 to 10 metric tons per hectare. Where in California, you can get 100 to 110 easily. What is really sustainable in terms of producing food that is going to feed the growing population? So for the emerging middle class, bottom line is it's the same as the rest of the world. There isn't, I don't see there's anything specific for that. Because local processors are driving the processing capability. What sort of stainless steel plant are they going to build? Are they going to try to peel? Are they going to make paste? What are their specifications? You're still marketing for them. And until somebody puts up a processing plant, there's no point growing processing tomatoes. So, and for us, we look at some specifics for tomato paste. That's international markets. We look at soluble solids and juice viscosity. Those are 2 very important traits. Okay? This is tomato juice. This is a very high viscous variety. These are forks, by the way. And this is a very runny variety. As you might imagine, even when I ask 3rd graders, they always pick this is better for ketchup. If you're making ketchup out of this, it's not going to be nearly as good. Are you doing more regional products like peel and dice, something like this? You look at the quality characteristics. And you have to have adaptability. So you need to, are you in a humid climate, like Ohio? Are you in an arid climate? You need to have the trialing system, you need mechanization, you need to understand the insect challenges and the disease challenges. So it's a matter of going from the research, that's great, I mean that's what we focus on, but you really have to do the full circle. You have to get it out into growers' fields and a lot of people following you around. That was a joke too. A lot of people, these are our dealers from the European area. They were looking at some trials because this was actually one of our grower trials. and you have to be aware, you have to get that into the factory. All of those places have to. So, that's my presentation. I have 2 last slides. One is I have to put in a plug for a UC Davis Solanaceous conference coming up in September. And so this is not going to be just talking about tomatoes, but overall, solanaceous crops. Alan Van Dynes and Ann Powell are the 2 leads on that, so they asked me to put in a plug. And finally, as a segue into Allison's talk after lunch, so Breeders' Digest used to have these little things, and what plants for something red and round that makes use? Meatball. So, but it's still got the horns, Allison, so we're not quite there yet. So, anyway, thank you again for your time, and I'm sorry if I went a little bit over.